Now we're going to go over a genetic cross. So they're always, 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 always going to ask a genetic cross in every paper. It just depends what. Like, are they going to ask you to draw it? Are they going to ask you to interpret it? There's like a small chance they'll ask you to do a dihybrid cross, but surely not. I'm not going to go over that. Well, not in this video in any way. I mean, I'll go over it by myself, but oh, doing maths and life science, it's like in paper one, 3 marks to work out because I literally just did 0,028 times 25 and I got like 0,7 milligrams. Like that was it milligrams or I think it was milliliters. How is that 3 marks? Like I wish math was no. Let's not talk about maths. That's over with. But if math was that easy, I don't see what three. I mean, I think they. I don't know. I don't know if there's a formula for that because I didn't use the formula. If there is, anyways. So this is an exact layout. You get marks for this proper layout. There's another layout to do, which I don't do, and that's not my problem. This one's the easiest one. So so this is where I said the phenotype and the genotype would go in. So there's some things you just simply have to write, which is this. The P1, F1, always have to write it. The meiosis and fertilization, always have to write it. Some of it seems useless, but that is cool. So do I say, yeah. So P1 is the parent generation, so your parents, and then F1 is the family, so the kids. That's basically what you're doing when you're working out the Punnett square. You're working out what the chances are of the different characteristics of the children of these parents. So we use capitals for dominant and we use small letters for recessive. Like I said, big B, like a capital B is dominant and a small b is recessive. That's the Punnett square. I'm sure we know how to do it. You put one gamete on the top, one gamete on the side, and then you mix it around. It's not that difficult. I think I swap phenotype and genotype sometimes. They won't mark you down on it. They could ask you for the ratio or percentage. My teacher just told us to do the ratio and percentage just in case, you know. So just make sure that you do if they ask for the percentage, give the percentage. Um, like some things you just give. Otherwise, at the end you just list all of them if they don't ask you for either. Monohybrid is one. Okay, well, mono means one, so that's one characteristic. And dihybrid is two because di means two. So I'm not going to do a video on a dihybrid cross because I was only just taught it like recently with the foil method and everything. It's it's an unlikely scenario that they're going to ask you the dihybrid cross. So I wouldn't give it too much stress if you can't do it or you're worried about it. It's the foil method. But if you do this layout, even if you don't do the gametes right, you should still get 1, 2, 3, like 3 marks out of the I don't know how many marks it would be, but you should still get the 3 marks because you'll get 1 mark for saying P1 and F1. You'll get 1 mark for saying um ho fertilization and meiosis. And then I think you'll get a mark for your phenotype and a mark for your genotype. So you might actually get like 4 marks, even if you get it wrong in a dihybrid cross. So it's not that deep. So yeah, I'm just doing this on a sticky note because I feel like it's important. It's an important concept to go over, which is incomplete dominance and codominant. So complete dominance is what we already know, where dominance masks the recessive, right? Incomplete dominance is when two phenotypically, physically different parents' alleles are not dominant or recessive. So they blend to make a new phenotype. So I drew a little example down there for you. It'll make sense once I color it in. But basically, they use flowers for most examples. Don't ask me why. But basically, the way I think of it is that it's neither dominant or recessive. So it's incomplete. So it's like there's no answer. So it's like incomplete because it's neither. And neither is like not an answer. So it's incomplete, if that makes sense to you. Um yeah, so you just need to keep this in mind if they ask you to do a genetic cross and they tell you it's incomplete, then all the gametes of the children will be the same blend of phenotype because it'll take one from each parent and just be the blend. So there in the example, you can see a blue flower plus a pink flower would be a purple flower because the blue is not dominant or recessive and the pink is not dominant or recessive. So they combine. And when you combine blue with pink, you get purple, basically. I hope the way I explain incomplete makes sense, like how to remember it, because you need to know the difference between incomplete, dominant, and codominant. So the way I explain codominance is that so they're two phenotypically different parents, but both their alleles are equally dominant. So they're both dominant. So the way I think about it is this is so convoluted, but the way I think about it is if you have divorced parents and they're co-parents, then they're both dominant, but they're both your parents. So then if you have a pink parent and a blue parent, then you'll just have a pink and blue parent. I feel like that made sense. The I feel like in theory it makes sense to me because co-parenting means that both of your parents like parent you, they're like responsible for you like 50-50. So that's basically what this codominance is because the blue flower will mix with the pink flower and just become a blue and pink flower. Like it'll have spots on it. So yeah. So the allele will be expressed equally in the phenotype of the offspring. So you need to also keep this in mind in a genetic cross because they use in the textbook they used cows. So they crossed what did they say? Like a red cow and a white cow. Red in this instance actually means like brown, but they say red. So red and a white cow, and then you get a baby cow that's white but has red patches on it. That's the real life example. Yeah, I'm just saying that the blood group AB is an example of codominance. I'll go into that more when I actually look at blood groups, but that's an example of codominance because the blood group A is a blood group and the blood group B is a blood group, but together they're also a blood group, which is AB. So it's not like it masks the other. So it's not like A is dominant over B or B is dominant over A. They're both dominant. So it's AB.